Muy bien, continuamos y es también un honor para mí el presentar a la doctora Sharon Hillier. Sharon Hillier es doctora en microbiología en la Universidad de Pittsburgh y profesora de enfermedades infecciosas en medicina reproductiva. Durante toda su trayectoria, el trabajo de la doctora Hillier se ha centrado en la prevención, en el diseño y en la evaluación de estrategias de prevención para la transmisión del VIH. Quizá me gustaría destacar que los trabajos de la doctora Hillier han asumido un reto realmente importante, ¿no?, que es la transmisión, el ingeniar procedimientos para evitar la transmisión del VIH a través de mucosas, fundamentalmente centrado en mujeres y especialmente en ambientes donde, bien por la falta de intimidad, bien por el estigma, algunas de las medidas preventivas que han tenido éxito en algunos otros grupos, ¿no?, realmente han conseguido resultados muy limitados. Ella es actualmente la coordinadora de un gran esfuerzo internacional para la evaluación de todas estas estrategias, que supongo que será el motivo de su charla, que es la red de ensayos clínicos de microbicidas. So it's an honor to introduce Dr. Sharon Hillier. The title of her talk would be Prevention, Prevention, Prevention. So thank you very much.
Thank you. Again, it's a real pleasure not only to be with all of you tonight. It's been a wonderful experience to experience San Sebastian, surely one of the most beautiful cities I've ever visited, but also to follow my wonderful colleague, Dan Kuritzkis, and his brilliant presentation. So we'll end tonight with a few thoughts about uninfected people and how we can strive to keep uninfected people healthy. And it's safe to say that it's been in a remarkable last 10 years when so many trials have been conducted, and we've seen both some amazing successes and some really huge disappointments. So I'm going to start with a summary of completed trials. I'll spend a few minutes on oral pre-exposure prophylaxis with tenofovir emtricitabine. I'll talk a bit about the dapivirine vaginal ring that's been evaluated for prevention in women. I'll talk a little about long-acting injectable ARVs that are being evaluated for prevention of HIV, the use of broadly neutralizing antibodies that were just mentioned by Dr. Kuritzkis in treatment, which are now being applied for prevention, and the new HIV vaccine trial that was just launched yesterday, building on the success of RV144. So in this slide, I've encapsulated a decade of research with nearly 50,000 participants. I don't know how many millions of dollars, but really a remarkable decade. It started with the 3 trials of male circumcision, the one in Orange Farm, South Africa, the Kisumu, Kenya study, and the Rakai trial, each of which showed a 50 to 60% reduction in HIV, and surely this is one of the most sustainable and effective single prevention modalities we have today. There have been 3 completed vaccine trials in 2007, 2009, and 2013. The Merck vaccine, which was HVTN 520, was shown no effect. The ALVAC AIDS-VAC RV144 study, which I'll talk more about in a little while, showed a 31% benefit, which did not quite reach statistical significance, except in the overall intent to treat, but not in the per protocol analysis. And then finally, the AD5 DNA prime boost study, the vaccine step trial, that showed, a 0% effectiveness. And the next, I've just lit up all the oral PrEP studies, and there have been a total of 8. The IPREX study, which was done in men who have sex with men, showing a 44% reduction. The Botswana study, published in 2012, showing a 60% reduction. The FemPrep trial, though, showing no reduction in young women with an average age of 23. The Partners PrEP study, published that same year, however, in Sarah discordant couple, showed a 67% reduction in HIV in when women and men taking oral tenofovir, and a 75% reduction in those taking tenofovir emtricitabine. This drug combination, or tenofovir by itself, was shown to reduce HIV in intravenous drug users in 2013. And then the Ipergase study, I think, and the PROUD trial, which each demonstrated that in men who have sex with men, reductions of HIV in over 86% could be achieved. Then there were 3 trials of tenofovir-containing gel. The first by Slim Abdul Karim and his wife Karacia, published in 2010, showing an exciting 39% reduction, but 2 subsequent trials from 2015 showed no benefit. Importantly, treatment is prevention. The HPTN 052 study showing us what we all knew, which is that if one achieves good viral control, that transmission can be virtually eliminated. And then finally, 2 vaginal ring studies, each showing about a 30% reduction. When you look at this scorecard, it's an amazing testament to how many people and how many groups have worked hard to really identify different methods to prevent HIV. Although it's easy to become complacent because of the amazing successes with treatment, we have to always remind ourselves that for every 2 people who go on treatment, 3 more become infected worldwide, and therefore we really have to have new methods to prevent HIV. So I'd like to spend some time now talking about where we are today with topical microbicides, PrEP, and vaccines. As I've just summarized, there have been 8 clinical trials of tenofovir-based oral pre-exposure prophylaxis, and 6 of the 8 showed reduction in HIV. Importantly, the 2 that did not were conducted in young women where adherence was low. This drug is now FDA approved, and there are many, many demonstration projects on oral PrEP underway globally. There were 3 completed trials of tenofovir gel, 1 of 3 positive, 2 completed phase 3 trials of dapivirine ring, which will both be published in a couple of days in the New England Journal of Medicine. Integrase inhibitors are now entering into the prevention space, and phase 1 studies have been completed, and a cabotegravir phase 3 study is set to launch. There are also large studies of infused monoclonal antibodies, the VRC01 antibody that Dr. Kariskas just mentioned, underway, and 3 HIV vaccine studies have been completed, none statistically significantly protective, but as I mentioned, HVTN 702 launched yesterday. So let's spend a little bit of time on each of the 3 groups that I'll talk about: the notion of oral PrEP and the dapivirine ring. So when we think about those 2, here's the dapivirine ring on the left-hand side and 30 days of oral PrEP on the right. Oral PrEP is now approved. The dapivirine ring trials have been completed, and the regulatory dossier is being submitted. And I think for the first time, we might begin to see options for women and men in terms of using ARVs. As I mentioned, the PrEP studies taught us, very importantly, that ARVs work only if they're used, and that younger people and people of color, people who are just entering their time of sexual activity, really struggled with adherence to daily oral PrEP. In this slide, what you see summarized is the reduction in risk of HIV, and that's graphed against the percent of people having drug in their blood when they were evaluated. And what you can see is that the higher the proportion of people who had adherence to the product, the greater the reduction in HIV risk. And this was from a nice summary just published by Jenner Fauner in AIDS just a couple months ago. Now, these 2 studies, the IPREX trial done in men who have sex with men and the Partners PrEP study, were selected from amongst those 8 to lead to the FDA licensure. And I think the continued body of evidence showing that oral PrEP works when used led to the guidelines published this year by the World Health Organization, stating that oral pre-exposure prophylaxis containing tenofovir should be offered as a prevention choice for people at substantial risk of HIV. And substantial risk really means 3 infections per 100, so a 3% incidence per year. In the US, and my guess, in Spain as well, that really generally refers to men having sex with men, and particularly receptive anal sex, who are not currently using condoms. But I think this guidance from the WHO has really put oral PrEP out front and center, and we understand we have much to do to implement oral PrEP. Now let me spend a minute or two on vaginal rings. The idea about vaginal rings was to try to address the problems we've seen with daily oral PrEP in young women. These vaginal rings are longer acting. They're used monthly, so inserted by the woman herself, and they provide very low levels of drug over a month. The women can insert it and remove it themselves. There's little or no impact on sexual activity. And because the amount of drug released over the month is so small, only 4 milligrams, the safety profile is quite amazing. No need to monitor creatinine. Because the rings can be inserted by women, they can be removed in private, they really do provide a somewhat stealth option for HIV prevention. Vaginal rings are available, as many of you know, for contraception, both in the US and in Europe, and these rings are very similar to those. It's a flexible silicone ring. It was developed by the International Partnership for Microbicides, and it releases, as I mentioned, a very low level of the drug dapivirine. This ring was evaluated in 2 phase 3 clinical trials, the ASPIRE trial, which we were involved with, as well as the RING study. These 2 studies included 4,588 women in 4 countries, South Africa, Uganda, Zimbabwe, and Malawi. And in these studies, the women had an average age of about 26 years of age. Most were unmarried. What we saw overall was somewhat disappointing. In the ASPIRE trial, we saw in the intent to treat analysis a 27% reduction in HIV. And in the RING trial, they saw a 30% reduction. But when we looked in the intent to treat and just did a pre-specified analysis of younger women, what we found was that there was an overall 60% reduction in HIV among women greater than 25 years of age. Why? Because younger women were more likely to take it out. When we looked again at women between the ages of 21 to 25, we also found that for the first time we had a product that worked pretty well in 22 to 26-year-olds with a reduction of about 60%. So the real problem, or the problem with non-adherence, was observed in 18 to 21-year-olds. So it really became important to understand, did this ring work if it was used well? So how have we gone about doing that? We collected blood from all the women in the study at every quarterly visit, and every month we collected the rings that the women brought back. And those rings were ground up, and we could extract how much drug was left over. Dapivirine is a very hydrophobic drug and won't come out of the ring unless it's placed in a fat-rich environment like vaginal fluid. And so what we were able to do is figure out by subtracting how much drug was loaded into the ring by how much was left at the end, how well the women used the product. And we were able to look at that interval of time before when the women used their ring before they seroconverted. Now this is the actual distribution of drug levels we saw in the rings when they were returned. And what you can see is that most of their rings were loaded with 25 milligrams. And what we decided was that we would say if at least 3 milligrams came out of a ring, that it had been used. So in women who had between 22 and 16 milligrams left in the ring, we judged that to be medium to high adherence. And in those who had more than greater than 22 milligrams left in the ring, were defined as having no or low adherence. So then we decided to look at how well did ring use and adherence match up to protective benefit. The HIV incidence in our placebo group was 4.7% per year. And so what you can see is in the most adherent group, the incidence was 1.2% or 1.6%, which is a 66 to 75% efficacy. And for those with low or no adherence, we saw obviously incidence that was much higher of 2.7 and 4.9. In other words, not surprisingly, ARVs work if you use them, but the ring has to be in place for it to work. And what we found is that the youngest women were taking the ring out. Well, why would they do that? In interviews we've conducted with the young women since we completed the study, we found that younger women had more worries. They were concerned. There was also some, I think, defiance and desire to essentially fool the study teams. It's been very interesting as we've gone back and talked to the young women who were in the study that it was widely understood among the very youngest women that they were gaming the system in a way. So we are going back now and beginning to understand how to engage more actively with the youngest women and are really taking a great many lessons from the contraception world in which we've done a lot of engagement about what young women want. So what we found was that the ring was extraordinarily safe and effective in women greater than 21 years of age, and protection was obviously quite increased among those who used it consistently. Overall, even though there was a lot of non-adherence, we still found much better adherence with this vaginal ring than we ever observed in young women using oral PrEP. And that with continued use and counseling, the young women's worries about the rings reduced, and with that, we got better adherence. We found young women were able to insert and remove the ring on their own, and it did not interfere with sexual activity. And those who talked to their partners about their rings actually found they could use it better. So we found across multiple different analyses that there was really an important relationship between ring use and HIV protection. It was not 100%. We think it's about 75%, and so it's a platform on which we can build. What we did find is that for some young women, the ring was quite empowering in a way that we never saw with the vaginal gels or with the oral products. This is a quote from a young woman in South Africa. She said, "I like that the ring stays inside you and no one can see it. You don't have to disclose it to others if you want. My family doesn't know I'm using the ring, and my partner can't feel it as well." And we found that with time, young women actually got to really feel that this was something that belonged to them. So moving on from oral PrEP and the rings to long-acting injectables, I'm going to talk about cabotegravir and very briefly about rilpivirine. Now, a lot of people have been excited about long-acting injectables because the idea is you wouldn't have to worry about all this adherence. You wouldn't have to worry about taking a pill every day or if women took their rings out of their vagina. There has been two completed studies of rilpivirine, and both of those studies actually showed that drug levels were much higher in the gut than in the reproductive tract, something we've also seen with tenofovir. And we also saw with rilpivirine that even a year after the last dose of a single dose of rilpivirine, that there was still an NRTI, rilpivirine, there in the plasma. With cabotegravir, there have been two phase 2 studies, HBTN 077, which included 200 men and women with 8 sites, and HBTN 076 with rilpivirine of TMC 278, and I believe these may be reported very soon. And then there's one completed study in MSM called the ECLAIR trial, which I'd like to talk about for just a minute. So in the ECLAIR study, there were 105 volunteers, all gay men, aged 18 to 65, defined as being at low risk of HIV, given their behavior, and they were given 3 intramuscular injections of cabotegravir 12 weeks apart. And again, that was based on animal model data suggesting that's the level of dosing that would be needed to protect. 21 other volunteers were given placebo, so it was a 3-to-1 randomization, and there were no safety concerns and no serious drug-related adverse effects. But just as animal model studies have failed to predict efficacy in some of the therapeutic vaccines, they've also been quite monkeys we've decided really aren't that honest with what they teach us in research. They also failed to predict correct dosing for cabotegravir. And what they found that was different was that cabotegravir reached higher peak levels faster, but was also eliminated faster from the body than expected, except in those with a higher body mass index, where they reached their peak slower and eliminated the drug more slowly. So there were two effects from this. One is they decided they couldn't give cabotegravir every 3 months, they would have to give it every 2 months, but there was also the tail problem. So in this figure, what I have is the proportion of people in the Hillier study who have drug after their last injection, and then on the bottom axis, the months since the last injection. And so what you can see is at 3 months, only 3% of people had no drug detected. By 6 months, 35%. By 9 months, 69%. And by 12 months, 84%. But what that means is that there's 16% of people, even after a year, who still have drugs present. The red bars are what we are calling the resistance zone. This is less than the effective concentration in tissue. And so what you see is that you have subtherapeutic levels that are present at concentrations not high enough to be effective, but high enough to potentially induce resistance. So you can see that was 12% at 3 months, 41% at 6 months, 22% at 9 months, and 16% even a year later. And then finally, the proportion that had therapeutic levels dropped from 85 to 24 to 9. So what does that mean? That red resistance zone is what we worry about, because if we want to use an injectable because it will enhance adherence, how do we manage when people have had their last injection if they go and become infected with HIV, having subtherapeutic levels of drug? And this is called the drug tail. So these injectable drugs have a long and somewhat unpredictable tail, and unpredictable because higher BMI is somewhat associated with a longer tail. And during these tail periods, if people become HIV infected, they could be at increased or greater risk of having a resistant virus to the integrase inhibitors, which would be somewhat catastrophic. So one of the possible solutions is to provide oral PrEP until the tail is gone, or to identify more reversible strategies, such as making implants or something like that that could be removed once people decide to come off PrEP. This has really been a struggle as we think about how to design trials using injectable PrEP. This is the outline or the schematic of how the HPTN 083 study of injectable cabotegravir is designed. This is a study that was set to launch actually about now. And you can see here that they were going to include 4,500 MSM and transgender women in North and South America as well as Asia. And how they were going to manage the tail is by providing an open label Truvada for 1 year following the completion of the injectable phase. So what's happened now that the cabotegravir data has come from Hillier? Essentially what they're doing or proposing now is to extend the follow-up with the oral PrEP for quite a long, about 6 more months. So this is I think the challenge as we think about high adherence injectable forms or sustained delivery is how do we manage that? So if we think about those kinds of ARV-based prevention products with oral PrEP, what we have is overall lots of green checks, very good, but it does require creatinine testing. When we think about adherence, the monthly vaginal ring or the cabotegravir injectable, that's really good. With oral PrEP, you do have to do medicalized monitoring, especially for creatinine, which was not necessary and won't be necessary with the vaginal ring. We don't know yet about cabotegravir, but I don't think so. A real downside of the vaginal ring is that it won't protect against anal sex, transmission during anal sex, because the drug levels that are in the rectum are quite low. We don't know yet for cabotegravir, but the main gap that we see in cabotegravir is this very long and somewhat unpredictable tail that would have to be managed with alternative PrEP strategies. So no one of these single methods is likely to be perfect. Each is going to have to be weighed for efficacy and how it can be most carefully rolled out. And so finally, let me spend my last few minutes on broadly neutralizing antibodies and vaccines. You probably saw these announcements about NIH launching these large clinical trials of antibody-based prevention. These are called the AMP studies, and they're studying exactly the same antibody that Dr. Kritzkis mentioned in his presentation, where they used it as a therapeutic, withdrew the ARVs, and then saw some rebound. A real question is, will these antibodies be able to protect against HIV in an uninfected person? Now, these are complicated studies. Volunteers are coming to the clinic and actually having IV infusion of antibody. So there are two of these. There's one called HVTN 704 and one called 703, and they're both phase 2B studies with several thousand people, and essentially evaluating, one is in men who have sex with men and transgenders, and the other is in women. The objective of these studies is to determine whether and how the VRC01 broadly neutralizing antibody can prevent HIV. And this will help to develop a marker that correlates with the level of protection and could provide really important insights into the correlates of protection. If it works, it could really be important in informing the vaccine agenda by defining whether or not antibody can provide protection, and it could provide immunogenicity study endpoints in phase 1-2 trials when they look at candidate vaccines. It's unclear at this time whether or not this product will actually become a product for prevention, but it is an important precursor to some of the vaccine studies, in as much as it will inform or have a more rational approach for the antibody-based vaccines. And finally, the HIV vaccine trials to date. As I mentioned, there was a 2007 study that was based on the T-cell vaccine approach. It was the Merck vaccine. And there were two studies, STEP and Fambili, both which had actually a paradoxical increased risk of HIV, not reaching statistical significance in the STEP trial, but did reach statistical significance in the Fambili study. And that was really thought to be due to the immune activation related to the adenovirus vector that actually enhanced susceptibility to HIV. At the same time, the RV144 study of was being conducted in Thailand of ALVAC, the canary pox and AIDS vax, which actually showed a 31% decrease in HIV. And then the third trial, as I mentioned, HVTN 505, the VRC DNA prime adenovirus boost, again, with no protective benefit. What happened after the RV144 study is that about a huge number of studies were undertaken to understand if the modest benefit that was observed in that study was real. And so they went back and did many, many secondary analyses. And what they found from these secondary analyses was with the IgG antibodies against the V1, V2 region of the envelope protein was associated with reduced infection. So they could localize which antibody was important. They found that non-neutralizing antibodies mediated the ADCC activity, and that the IgA antibodies were actually correlated with increased infection. So based on that, The RV144 vaccine was modified to include clade C, which is the clade found in South Africa. They used a different adjuvant, MF59, and they're proposing using booster injections annually. The study will enroll 5,400 men and women ages 18 to 35. It's being done in Africa and funded by the United States NIH, the Bill and Melinda Gates Foundation, and the South African Medical Research Council. And at least in the press release I read today, they say that results are expected in 2020, but you never know. They can go upside down or they can go faster or slower. This is the press release that was put out yesterday. This is the first new HIV vaccine efficacy study that's been launched in 7 years, and I think all of us are rooting for a success, and we certainly hope that this new strategy does show some evidence so that it can lead the vaccine field forward. But as a researcher who has worked in this area for more than 20 years now, I can say that I have learned that no single thing, there will be no single magic bullet that actually works for prevention. I believe for some people, a tablet using tenofovir, emtricitabine, or maybe potentially even Maraviroc is going to be perfect. And here in Spain, where your epidemic is so much driven by men who have sex with men, I think rollout of PrEP is going to be really important. For women, I think vaginal rings will play an important role because it's less medicalized, requires less medical monitoring, and vaginal rings, including dapivirine, have completed trials, but there are also 90-day tenofovir rings that are entering testing, tenofovir disoproxil fumarate, Maraviroc, Vicroviroc, and another integrase inhibitor called MK-2048 that have all completed phase 1 studies. Injectables could in fact be an important progress forward if in fact we learn how to manage the tail. And I think that's going to be the really important and challenging part of the injectable venue. There's also new products that are being developed for men who have sex with men and women who practice anal receptive sex, including inserts containing integrase inhibitors like elvitegravir, tenofovir, and TAF that are entering phase 1 studies. At Pittsburgh, we're also developing quick dissolving vaginal films, and I've mentioned as well vaccines. What we've learned is that different people will want different things, and if we have a broad armamentarium, we can find products that can fit into people's lives and can provide them the power to protect themselves from HIV. So in this very quick tour through the world of HIV prevention, I hope you'll agree with me. There's been breathtaking progress over the past decade. We need to see broad implementation of oral pre-exposure prophylaxis to all groups who need it. Looking ahead, we think vaginal rings may be approved by regulatory authorities in 2018 or 2019, and there's a lot of enthusiasm for next generation pre-exposure prophylaxis using a range of new formulations of ARV. And finally, HIV vaccines are new again with the first new vaccine trial just being launched at the same time that we're studying monoclonal antibodies, which could potentially provide insights about neutralizing antibodies and prevention of HIV infection. So I hope that I can stand here, because I'd like to come back to San Sebastian in a few years and say, instead of all this work going on, that we actually have more success, that we see broad implementation of prevention, that we see progress in cure and better treatments that require less daily adherence, and that we can really have a hope of an AIDS-free generation. Thank you so much.